Hello, everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the 3 NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. I am joined today by my colleague from NCCAT, Ed Eng, along with Kirsten Wheeler from the Pacific Northwest Cryo-EM Center, PNCC, and Michael Schmidt from the Stanford SLAC Cryo-EM Center, S2C2. S2C2 is hosting today's speaker, Virender Singh, who's joining us from the Center for Alzheimer's and Neurodegenerative Diseases at UT Southwestern Medical Center in Dallas. And Mike will introduce him after a quick update from each of the centers to keep you up to date on the resources we make available at no cost to the research community. This is a regular event we host the last Thursday of each month to highlight methods and tips and tricks that groups are using to collect and interpret the data they can obtain with the help of the National Cryo-EM Center resources. You can already save the date for our talk in August. PNCC will be hosting Dr. Camden Driggers from OHSU, who'll be talking about KATP channels in metabolic sensing. And we'll have the registration link for that available on the events page at cryoemcenters.org in the next couple of days, so watch out for that. Before we get to today's business, if any of you are attending the microscopy and microanalysis meeting in Cleveland next week, so are we. So staff from all of the cryo-EM and cryo-ET centers will be there. So come find us, say hi, or if you have friends or colleagues you know are going, send them to us to say hi. And if you can't find us in various talks and poster sessions, we do have a booth in the exhibit hall, representation from the single particle focused and the tomography focus centers, so come say hi. Back to today. If you have questions during the talk, please send those using the Q&A box, not the chat, and Mike will be moderating questions at the end of the talk. So I'm going to turn over to our center representatives. Up first, Ed, let us know what's happening at NCCAT.
Hi, I'm Ed, coming from NCCAT. We offer access, screening, and training. We just commissioned our Glacios 3 here at NYSPC. That's the second Glacios for the National Center. It has a Selectris and Falcon 4i, so that'll bulk up our screening and training programs. Talking about M&M, the 3DM FIG will have 5 sessions, so if you want to be in room 12, you can see that. And please stay in room 12 for Thursday, because Claudia Lopez at PNCC and myself are organizing an EM and Education Symposium, and you'll have a lot of National Center representatives there. So if you want to find out more, please go to nccat.nyspc.org.
Thank you, Ed. And Kirsten, why don't you let us know what's happening at PNCC?
Hi, my name is Kirsten Wheeler. I am an administrative coordinator at Pacific Northwest Cryo-EM Center. PNCC specializes in a few different things, of which 2 of our training types and accessibility avenues are general access as well as screening access. Screening access is up to 5 days of scope time where up to two recipients are able to come on site. General access is actually a 2-year proposal where you can actually utilize the data collection as well as screening on our scopes. We have multiple scopes, of which we have 4 Krios with K3s as well as Falcon 4i cameras. We also are awaiting the allowance of our Glacios 2 microscope. That's our newest microscope, and also our other screening access or screening microscope is our Arctica. PNCC recently actually notified our general access users to be able to do our sample preparation workshop. We have multiple workshops that are available to our general access users. And then there's also another one that is our cryo-EM workshop that is open to the public. If you have interest in contacting PNCC for access, please go to our website.
All right, so now I will turn over to Mike. Mike, let us know what's going on at S2C2.
Hi, I'm Mike Schmidt. I'm one of the co-directors of the Stanford Slack Cryo Center, and we just started. We just finished accepting our running the acceptance tests on our fourth and final cryos. The engineers have been very busy, and they are moving on now to installing and commissioning our glacios. And after that, we'll go. They'll commission our tundra. So we'll have four trios, a glacios, and a tundra for our high resolution and screening and training scopes. Our next workshop will be very exciting. It's in September 16th to the 18th, and it will have in-person. Our in-person quota is filled, but you can join in by Zoom, where we will run a CryoSpark workshop with Ali Punjani and his colleagues who invented CryoSpark, and will give us a lot of tips about how to use CryoSpark efficiently. So join us either at the workshops or join us for data collection at Slack.
All right, I'll stop sharing, Mike, and let you introduce today's speaker.
Oh, it's my great pleasure to introduce Virender, and our collaboration with the Saelices lab is longstanding. She was one of the Lorena was one of the first people that made use of the three centers. Has been at least two of the centers for data collection. She has used our center extensively since she was a postdoc in David Eisenberg's lab at UCLA, and she moved on right after that to UT Southwestern, where she started a lab there. And we have hosted at this webinar Bin Nguyen from her lab 2 years ago. And Virender is continuing that line of research. It's very interesting research on amyloid fibers in another organ that's very hard working besides our brain, that is the heart. And he'll talk about amyloid fibers and what they can do to various organ systems. And I'm really interested in finding out. Okay, thank you, Virender.
Yeah, thanks a lot, Mike, for the introduction. So let me share my slides. Is it visible?
We are seeing your presenter mode. Just switch,
Okay, just
swap to play. Yeah.
Is it working fine now?
Yes, now we see your slides. Thank you.
Perfect. So yeah, I'm Virender Singh. Currently, I am postdoc at Saelices lab at UT Southwestern. And thanks a lot for giving me opportunity to present my work, which I have done for last 2.5 years. The work our lab mostly focuses on is on systemic amyloidosis, particularly transthyretin amyloidosis. And today I'll be presenting work which is related to revealing structural polymorphism in ATTR amyloidosis with insights from cryo-electron microscopy. So what is systemic amyloidosis? So it is a disorder that occurs when there is buildup of amyloid fibrils in different tissues and organs, mostly linked with systemic circulation. As we know, these amyloid fibrils are formed from well-ordered or intrinsically disordered protein, and under X-ray, so they show a particular pattern, which is cross-beta pattern with intra-sheet distance of 4.8 angstrom and intersheet distance of 10 to 12 angstrom. When it comes to systemic amyloidosis, there are more than 20 diseases which are linked due to the buildup of amyloids in different organs. And based on the protein which affect different organs, so it could either affect the heart, which is very common when it comes to light chain amyloidosis and ATTR amyloidosis. But there are many other proteins which only affects either kidney, few affects only liver. So, and each of these protein, based on the tissue they affect, they have different clinical manifestations. And many of these diseases have overlapping clinical manifestations, which make it difficult to diagnose. So what does systemic, or like, why does systemic amyloidosis develop? There are multiple factors when it comes to the reason behind the development of this. There could be intrinsic instability of the native protein. There could be clonal somatic mutations. There could be hereditary mutations, or it could be result of increased synthesis of proteolytic cleave products of that protein. All of these combined or in isolation could result in increased supply of precursor proteins, which then unfold, form oligomers or protofilaments in combination of multiple partners, one of which is serum amyloid protein, which binds to the amyloids in systemic circulation. They results in proteotoxicity toxicity and could accumulate in different organs and result in different clinical manifestations. One of these is cardiac amyloidosis, as Mike mentioned. So many of us know about Alzheimer's disease, where neuron or brain is the affected organ. But in cardiac amyloidosis, it is the heart which is most affected, and patients die due to heart attack because the heart muscles or cardiomyocytes are stiff and they cannot pump the blood the way they should do in under normal conditions. When it comes to cardiac amyloidosis, which is referred to again, buildup of amyloid proteins in heart muscles, it is mostly linked to light chain amyloidosis and transthyretin amyloidosis. Today, I'll be talking about transthyretin amyloidosis, but in future, our lab is also working on light chain amyloidosis, and maybe you'll see updates from it from other colleagues. Transthyretin amyloidosis, as name suggests, transthyretin is a protein which transport thyroxine and retinol binding protein in both blood and CSF. 90% of the transthyretin is produced in liver, but 2 to 10% is also produced by eye, mostly retinal cells, or choroid plexus. So under normal conditions, so transthyretin, which occurs as a tetramer, is composed of 4 subunits, and as I mentioned, mostly synthesized and assembled in liver. It performs multiple functions and undergoes clearance, and that's how the cycle goes on. But either due to aging or due to certain mutations, so the tetramer become unstable. It dissociates into monomers, which are not stable enough and results in amyloid formation. And this amyloid deposit in different organs, and that leads to different clinical manifestations. Either it could be cardiomyopathy, polyneuropathy, or there could be multiple or combination of multiple manifestations. When it comes to the distribution of ATTR genotypes, so there are different mutations which are linked to different geographical regions. Wild type is the most common kind of ATTR amyloidosis, which accounts for more than 50% of cases. But Beyond that, when it comes to the mutant transthyretin, V30M is the most prevalent, which occurs again in different geographical regions. And based on the geographical region, so the clinical presentation is also different. When it comes to US, so most of the cases are accounted by either T60A or V122I. But today I'll be focusing on I84S, which is a unique mutation, and I'll tell about it in more detail in coming slides. As I mentioned, so this is a systemic amyloidosis, and multiple organs are affected, but most of the clinical presentation are either defined as related to cardiomyopathy or polyneuropathy. ATTR wild type, which is again linked to the aging, is mostly considered a cardiomyopathy, whereas certain mutations, and I84S is one of them, is linked to polyneuropathy. But despite this classification, so multiple organs could be involved. There could be amyloid deposits in different organs, and there will be a mixed set of cases where the person might have both combination of cardiomyopathy as well as polyneuropathy. As I mentioned, so there are like two classes. One is wild type ATTR, which is mostly related to aging, and it has been observed that ATTR wild type amyloid exists in 25% of people above 80 years of age. So this is the data which is collected based on the analysis of tissues at autopsies. And it has been found that the ATTR wild type amyloidosis cases has more consistent clinical presentation. But when it comes to hereditary ATTR or variant ATTR amyloidosis, there are more than 140 mutations which could be linked to it. And the onset of this hereditary ATTR is widespread. It starts from 30 years to almost 70 years of age. And these represent a diverse clinical presentation when it comes to the symptoms or different organ involvement. So currently our lab is heavily using cryo-EM based on the published report, starting with the tau amyloid structure from 2017 to the validation of those structures using cryo-ET in 2024. So we now know that cryo-EM can definitely be used to study amyloidosis. And so most of the structures which were solved were from localized amyloidosis as tau or A beta, but our lab or our group is one of the group which is mostly focused on systemic amyloidosis. And till now, we have almost solved more than 25 structures from different organs or from patients with different mutations. So this is the protocol which we follow for the preparation of amyloids when it comes to systemic amyloidosis. We mostly use around 150 to 250 mg of tissue. It could come from fresh frozen hurt, or it could be a different tissue. So we mostly follow a water-based extraction protocol, which is mixing of the homogenized or minced tissue, washing it with water or a tris-based buffer multiple times, treating it with collagenase, which enables the loosening up of the amyloids, which are trapped in collagenase or which are trapped in collagen when it comes to hurt tissue. multiple washing with tris buffer, and then extracting it with ice cold water supplied with EDT or EGTA. So we are using chelating agent to get rid of proteins like SAP, as I mentioned, and those proteins get dissociated when the calcium which is bound to SAP is removed. At the end, we always check the quality and distribution of the fibrils using negative staining, followed by screening of the cryo-EM grids using Artica. And most of the data collection, we use the national centers. We heavily use the S2C2. I think currently for 90% of the data collection, we are depending on them. But for the data I'm presenting today, we have actually used PNCC, NCAT, and UT Southwestern facility as well. So the question which we are asking is, can we use cryo-electron microscopy to reveal structural polymorphism in ATTR amyloids? And our working hypothesis is the phenotypic variations are related to structural variation in amyloid. When we started, so at the time there was one structure out from ATTRv30M mutation. By the time we were working on solving structure, so there was another structure from I with again the same V30M mutation. So the organ for the the organ for the TTR production is different in these two cases. Here it is produced by liver and deposited in heart. Here it is produced by retinal cells, and the amyloid is deposited in vitreous humor. The third structure which came out in 2022 is from ATTR wild type. Again, so deposited in heart. So by the time, so our lab was also working on solving structure. So we started with ATTR wild type, and we tried to use multiple patient just to map if there is any variation from patient to patient or any structural polymorphism when it comes to ATTR wild type amyloid structure. So here I'm showing data from 4 different patients, which are in like above 70 years of age at the time of tissue, like at the time when we got the tissue from these patients. All the 4 ATTR wild type showed fibril distribution, which is consistent. Majority of these fibrils were twisted and curvy, and with minor population of straight filaments, which were not used for the data processing. At this point, we mostly picked all the particles manually to get the 2D classes, and at the end, we mostly observed the similar morphology of the ATTR fibrils. As you can see, all the density maps looks quite similar, and these are very similar to the one which was reported earlier from wild type as well as V30M ATTR. So when we modeled this, so we can see the conformation which is obtained by the TTR wild type amyloid. It has the N-terminal fragment which starts from proline 11 to arginine 34, and a C-terminal fragment starting from leucine 58 to threonine 123. So the initial 10 amino acids were missing from the density map, as well as there is a fragment from 35 to 57 which is missing. So mostly these are like disordered, and we don't observe in the cryo-EM structures. Upon solving the structure from all 4 cases, we observed similar conformation, which is a closed conformation. And this closed conformation has a polar channel, and it is very consistent in all the 4 cases. We also observed the similar secondary structural composition for all the 4 patients, as well as the one structure which was solved by other group. So all 4 structures show a good homology with RMSD of just 1. This reveals that cardiac ATTR wild-type fibril structures are very similar, and this correlates well with the clinical presentation that most of the wild-type ATTR amyloidosis cases has a similar clinical presentation. But is there any relation of amyloid structural diversity versus phenotype? So as I mentioned, so our lab has actually solved multiple structures now. ATTR wild type is one of them, but there are multiple mutants for which structure is solved. And most of the structures show the closed conformation, and which looks very similar from patient to patient and from one mutation to other mutation. But we came across a mutation, I84S. The patients with these mutations falls under polyneuropathy category. So this mutation is very peculiar. So it was first reported in 1956. It is one of the first reported case for transthyretin amyloidosis. It has been mapped to Indiana Swiss descent, where someone from Swiss moved to Indiana in 1880s. This is very clinically distinct with manifestations which are related to neuroocular and cardiac. So in this particular mutation, so there is no renal involvement compared to many of the mutations like T60 where the kidney is also involved. Patients with these mutations, particularly males, no one survived beyond 63 years till now. Females survive longer than males for this particular mutation. When it comes to clinical presentation, so based on different studies, it has been found that the patients show carpal tunnel syndrome in early 30s, followed by polyneuropathy, vitreous opacities, and cardiomyopathy in 50s. And most of the death is due to cardiac manifestation. So I84S mutation is important because I84 residue is one of the important residue which is involved in hydrophobic interaction, and that stabilize the loop, which is again involved in T4 binding. The mutation I84S results in perturbed capping interactions, and this results in low T4 binding. Again, this mutation affects the TTR interaction with retinol binding protein, and it has been observed that there is 3-fold less binding with RBP if the isoleucine is mutated to serine. There is also evolutionary perspective to it. So there is I84 residue, which is absent when it comes to fishes, and there is no TTR RBP complex in this case. But for most of the terrestrial animals, so I84 residue is present, and we see the RBP TTR complex formation. So again, similar to ATTR wild type, we had 3 cases with ATTR I84S mutation. We extracted fibrils using the similar protocol, and we observed consistent morphology of fibrils when it comes to negative staining and cryo-EM. All majority of fibrils were twisted. Only a minor class was the state ones, which are not used for the data processing. So as we see for patient 1, we observed similar class with the closed gate conformation, but surprisingly, we observed a new polymorph, which is different from the one which was reported till now. So in this case, so this is an open gate conformation, which if you see closely, so there is mutation at this residue. So isoleucine is now mutated to serine. So in case of isoleucine, so there is an interaction which was holding this arm, but due to the mutation, now this arm is loose and there is a twist at residue glue 66, and the conformation is completely different. That leads to the different interaction in this area. So the polar channel is now open in this case, and this is like the first polymorph for ATTR, which is reported in this case. Again, surprisingly, so both these polymorphs are actually mapped back to the same fibril. That means there are layers which contain both the closed conformation as well as open conformation. So the fibrils are made up of both fragments with isoleucine as well as fragment with serine. At this point, so the resolution is not good enough there where we can actually see this particular residue, whether it is serine or isoleucine. But based on the mass spectrometry, we see almost 30% of these fibrils contains I84, but 60% of this contain mutant serine. Then we solved the structures for the other two patients as well, and we observed consistent closed conformation for those two other patients as well. But the another conformation or the polymorph which were present were slightly different from the one which we observed in patient 1. So in one case, so there was the arm which was shorter or broken, and in another case, so the arm was slightly inclined, which actually mimics the V30MI structure which was solved earlier, although the resolution was not good enough, so we could not model it to that extent. But this gives hint that maybe the polymorphism might not be just induced due to one mutation or one tissue which is producing the TTR, but it could be that different mutations could result in different polymorphic states. So as I showed earlier, so this is the mostly affected region which we observed in case of ATTR I84S. Based on the structural diversity till now, which we observed, so most of the cases with cardiomyopathy, we observed consistent closed gate conformation, as I mentioned. So most of the wild type cases has this conformation. Majority of the mutations have this closed gate conformation. And most of the structures reported till now falls under cardiomyopathy category. But when it comes to I84S, which falls under polyneuropathy, so those cases show both closed gate conformation, which is again consistent with the wild type. But along with that, now we observed different polymorphs, which we again named as broken or open gate or absent gate. So due to this peculiar mutation, which is ATTRs, we observed different polymorphic states. And based on the limited data we have till now, we could say that the structural variation which we see in I84S could be linked to the polyneuropathy. So we have to solve more structures from the polyneuropathy category to actually validate this point. But the homogeneity in case of amyloid structures, which we observed in ATTR wild type cases, so they are mostly categorized or linked to cardiomyopathy cases, and many other mutations like T60A or other mutations which are mostly linked to cardiomyopathy also see or show a consistent closed gate conformation. So I like to thank Saelices lab for the opportunity given to me for working on this interesting project, and our collaborators, Center for Alzheimer's and Neurodegenerative Diseases. Again, the facilities, we try to use all the national centers for data collection, donors and families who actually donated the tissues, and the funding agencies. I think I'll stop here and I'm open to questions.
Thank you, Virender. We'll encourage more questions, but I have one to start with. The I84S mutation leads to this polymorphism, and you said that the polymorphism actually can happen along the same fiber. One section will have the closed and one section will have the open. But are all of the proteins in the fiber the S mutation? And if there's only one gene copy, which would be just the serine, then that part would be open. Is it possible though that you could have a fiber that's all the S mutation, but still has both open and closed, just because it's an equilibrium between open and closed, even though the mutation is in all of the chains?
Yes. So most of the cases till now which we have worked are heterozygous. So they have copies of both wild type and mutant. And based on mass spectrometry, so in this particular, all these three cases, so almost like 35 to 40% is wild type, 60% is the mutant in these amyloids. It is very difficult to say at this point because we have all these structures in the resolution range of around 3.2 to 3.5. So we cannot exactly say whether the residue which we are seeing is isoleucine or serine, because the density is not that good. But definitely there could be fibrils where there could be only mutant present. There could be fibrils where the multiple layers which we are seeing are coming from only wild type. And there will be fibrils which are like which have like layers alternating with like wild type and the mutant. This, like, again, we think that the loss of isoleucine to serine is leading to this open conformation. But as you mentioned, there could be possible that there are still some interactions which might be like keeping it in equilibrium. Maybe solving more structure or like reaching to a resolution of maybe 2.3 or 2.2 angstrom might exactly show or like give answer to what you are asking at this point.
Yeah, great. If a little higher resolution to let you distinguish leucine from serine. Yeah, that'd be great. We do have a
Yeah.
question come.
Actually, can I interrupt for a second? Because I had a
Well,
question on the same topic.
oh,
I'm
Yeah.
curious how you're separating out these two classes in your processing. At what stage in your processing are you separating these out?
So these classes are separated at 3D classification. In 2Ds, we cannot like differentiate between these. And for this, like initially, like when I started working on this using a similar pipeline which we were using, we were actually getting only this class with lower minima at different points. And we were not sure the data quality was good. And we were not sure why we are actually getting minima at multiple points. So at that point, we tried to change strategy. We tried to like use a overlap of single layer during the extraction of this like filaments in 2D and 3D classes, and that resulted in, I think, more number of particles. And because like as you see, so there are multiple layers which could be like this closed conformation, this open conformation. So we could only observe this conformation when we changed our strategy and isolated each layer, and could reach or like could find this hidden class from the uh, closed
So is
conformation.
that a matter of like changing your box size during extraction?
Not really box size. So if we like in normal pipeline, we use I'll say interlayer difference of like three layers. So the way like we extract each layer. So mostly like we use overlap of three layers.
Mhm.
But in this particular case, we tried to almost isolate each single layer. So that resulted in like pulling out this particular class.
very
I'm not
interesting.
sure if I'm like exactly clear on this point.
No, you did. And I'll just interrupt with one more question, and then I'll let
Yes.
our audience ask questions. But what software are you using to process these?
Yeah, so I think all our processing pipeline is relying on Reliant. For this, like we use Reliant 3.1, but currently we are using Reliant 4.0. Till these data sets, we were manually picking particles. So I think our whole lab spent months in picking particles from this. But now we are using like training Topaz, and we are like mostly relying on auto picking now. So, and we are, I think, getting good structures out of auto picking. So almost saving months of manual labor. Yeah.
Thank you. Great. So we have some questions in the Q&A from Olga Kursky. Great work. Could you please comment on the status of type 2 ATTR fiber structures currently in the works in your lab? Do you expect the amyloid fold to be different in between type 1 and type 2?
So yeah, so I'll like maybe tell about type A and type B. Maybe that's what
Okay.
yeah, it's about. So most of the structures which I'm showing are coming from type A, and type A fibrils are consisting of full length transthyretin as well as fragmented transthyretin.
Okay.
Type B is only full length transthyretin. So there are no proteolytic fragments. And most of the cases with type B show straight filament morphology. So till now, I think we don't have a structure which is good enough to represent uh, uh, I'll say any of the conformation we see, but our lab is, I think, working on it, and in future you will see some updates on it.
Thank you. I think this goes to the question of Christina too. What kind of mask was used for the 3D classification to get those two classes? I think you kind of answered that the mask is, if it's a smaller mask, then you can split these things up rather than getting them averaged together. You see the two classes stand out. Yeah.
Right. So it's like if you see fibril in this manner, so normally if we try to like cover it with overlapping segments, so in those
Mhm.
cases we are actually missing out because this might be over representing or like merge like say it is not allowing the isolation of this kind of classes. But if we try to isolate each single class and then
Mhm.
do 3D classification, in those cases, we are successful in getting a separate class. And I
Mhm.
think if, as you mentioned, so if there are like cases where you see a fibrils like with just this conformation or this conformation, in those cases, we might not have to change any strategy because you see actually those classes clearly. But
Yes.
these are like sort of like nature has created it in a way it's very difficult to isolate it with normal pipeline.
Yeah, and another thing was in building these fibers, is it cooperative? Is that when you start a closed conformation, do you continue with closed conformations for a while, or if you start with open, they all become open because they're kind of cooperatively folding that way, or yeah, getting to that or conformation. Um, that would be an interesting thing too, to see what nucleates one form versus the other, and is it easier? Another question I had is that I think you mentioned it too. Every one of these that you see is one half of the fiber, and the other half is related by C2 symmetry, basically a twofold, right?
Yes.
And the twofold axis is roughly around the corner from the I-84S position, and they touch each other there.
Yes.
And I think you said this too. These things are mostly, or at least partly, uh, fragments only from these two sections, 11 to 30, and then 57 to 100, or 58 to 123. So are these generated by the proteasome and the degradation pathway, and they just got kind of build up and start forming fibers after they've been cleaved?
Uh, I'll say like there's still not 100% evidence when the proteolytic uh, cleavage is happening. But definitely like when you see these fibrils have classes where few of the classes has full length TTR.
Hmm.
That means this part which is disordered, but it's there. There'll be
Yeah.
layers where this disordered fragment is cleaved. And again, so the cleavage has like residues ranging from like something is cleaved at 55, something is cleaved at 40 residues. So the proteolytic cleavage sites are spread across that disordered region. We did like mass spectrometry to find residues where we see that cleavage. Actually, it's not just in the disordered region. So there are fragments which might be even cleaved here. So,
Hmm.
and that could be one of the reason that we are not hitting good resolution, even though the data set is good, because the layers are not exactly identical. So there are fragments where you see proteolytically cleaved. So we cannot actually build or get a good density map due to that heterogeneity.
Okay, thank you. We have, it's really fascinating.
Yeah.
We have another question. Thanks for the great presentation. Are you facing ferritin contamination similar to extraction of fibrils from brain? How are you solving that if in your preparation procedure?
So good question. So actually, my cryo-EM training actually started with tau extractions from brain, and I'm aware about the ferritin contamination from brain. Compared to brain, most of the like systemic amyloids, we don't see ferritin contamination. If you see the protocol which we are actually using even for extraction, it is very different from what we follow for tau or any brain derived. So we don't use any detergents. So these tissues are heavily loaded with these amyloids, and it's just getting rid of collagen, which is one thing. If we don't get rid, you don't get the isolated fibrils, so you cannot solve structure. Another thing is getting rid of serum amyloid protein, SAP. It decorates the amyloid fibrils, and the calcium is one key factor. So when calcium is bound to SAP, SAP bound to amyloid. So if you get rid of that calcium, so the SAP, which is a pentamer that disintegrates, and you see the fibrils, I think, more in soluble fraction compared to in pellet. So when it comes to systemic amyloid, I think this collagenase treatment is key, and use of EDT or EGTA is key. No issues of ferritin. If you see mass spectrometry data, so you definitely see multiple proteins coming from systemic circulation along with these amyloids, but they are not, I think, they don't harm or they don't give any issues when it comes to data collection.
Okay, one more question here. I was wondering what causes the blurriness in your 2D class averages in the background of the high resolution alignment. So those 2D are those 2D averages or those reconstructions, because you're seeing them from the top. Oh, okay, so those are the 2D averages that
Yes.
2D class.
So
Okay.
yeah, so these are and mostly like since I
And those
told
are
that
reconstructions, yeah.
These might be coming from this like the disordered regions which are not well ordered. So this all the fuzzy thing, and there are like few extra densities which still we couldn't figure out exactly what it is. Our lab is trying to work on recombinant transthyretin and using strategies to get structure which is more close to what we see from ex vivo fibrils, and maybe then we could ask more questions about different polymorphic states due to mutation, or what binding partners could be leading to different polymorphic states.
Any other questions?
Yeah, if we don't have more questions, we can wrap up today. Thank you everyone for joining us. We wish everyone well in their cryo-EM journeys. Hopefully they get as much amazing data as Virender has gotten. That was really impressive. And the national centers are here to support your science, so let us know how we can help you. And we hope to see you here again.